Welcome to solverfy.
So what can we do with metagenomics? Metagenomics is essentially a survey of all the DNA that can be found in an environment. So be it some ocean water, a soil, a tissue, or even when we start colonizing other planets, the soil of other planets to see if there's little bacteria there. So the first step in metagenomics is collecting the sample. The second step would be to isolate the DNA. The third step would be to amplify the DNA. And by amplify, you use polymerase chain reaction to make more copies of the DNA, because if you have more copies of the DNA, you can more easily sequence and align them. So after you amplify, you sequence the DNA, and then you use a computer to align sequences and compare. So sequence, alignment, and comparison. And then this leads to the identification of all the species that are in the sample that you've taken, and it can tell you if there are new species that are present in the soil that we haven't discovered before, or it can tell you the relative abundance of each species. If you have So because you're basically doing a survey, a direct survey of the environment, if there's a lot of DNA from one particular species, that tells you that that one species is the prevalent form of life in that particular environment. Okay, so that's the use of metagenomics. So the answer to this question is choice D. It identifies new species more rapidly and analyzing the effect of pollutants on the environment, because pollutants can affect the frequency or the abundance at which uh, organisms can be found, or the rate at which they can replicate, divide, if they are microorganisms. So if you compare, let's say, two different soil types, one where there's pollutants and one where there's not, if there's a lot more of one organism in the polluted versus non-polluted, or the other way around, then that tells you how the introduction of those pollutants is affecting the ecology of that soil sample. So choice D is the correct choice here.